This is part 2 for Chapter 17, Adaptive Immunity. Chapter 17 is all about your adaptive or specific immune response. And this is also known as your third line of defense. And your third line of defense involves the lymphocytes, the T cells and B cells, and the antibodies. And your adaptive or specific immune response is responsible for fighting a microbe that has infected your body. So it is a response to an actual infection. For your adaptive or specific immune response, there are two parts. The first part is the humoral immune response. And the humoral immune response involves mainly the antibodies and the B cells that make the antibodies. The second part of your adaptive or specific immune response involves the cell-mediated immune response, and this more specifically requires T cells. And the T cells will go out and kill eukaryotic cells. In the previous lecture, I talked about antibodies, and as a review, the basic structure for an antibody is a monomer, basically a Y structure, and it is composed of 4 protein chains, 2 heavy chains, and 2 light chains. And these are held together by disulfide bonds. And every chain has a constant region and a variable region. And the variable region of the light chain and variable region of the heavy chain make up the antigen binding site. And the antigen binding site has a particular structure that fits perfectly the epitope of an antigen. So when the antibody binds to the epitope of an antigen, we say the antibody is recognizing that antigen. I also went over the 5 classes of antibody. And the first class I went over was IgG. IgG is a basic monomer. It is found mainly in the blood, makes up 80% of the antibodies found in the blood. And it is so small that it can cross into tissues. So it can leave the circulation and go into the tissues. And an important part about IgG is that it is the second class of antibody made by an activated B cell. The next class of antibody I talked about was IgA. IgA is a dimer. It's 2 of those monomers bound together, and IgA is found mainly in mucus and other secretions such as breast milk. The main purpose of IgA is to prevent attachment. The next one was IgM. IgM is a really large antibody because it is made of 5 of those monomer structures. And IgM is found only in the blood because it cannot leave the circulation. It cannot leave the blood vessels and cross into the tissues. And IgM is responsible for transfusion reactions. The last 2, IgD and IgE, are monomers in structure, but they act as cell receptors. IgD is on the surface of B cells, and when it binds to its appropriate antigen, it serves to activate the B cells. IgE is found on the surface of mast cells and basophils. And mast cells and basophils, when the IgE binds to the appropriate antigen, it will serve to activate the mast cell and the basophil, and both of these will release histamine, which stimulates inflammation. So these are the 5 classes of antibody, and one particular B cell is able to produce all 5 classes. Then I went over the 5 results of antibody binding to antigen, or the 5 benefits of antibody binding to antigen. So the 5 things that will help fight an infection. The first result of antibody binding to antigen is agglutination. Because every antibody has at least 2 antigen binding sites, that means that mixing antibodies with the microbes, you will get clumping of the microbes. and this prevents the spread of the microbes through your body. IgM is really, really good at agglutination. The next result of antibody binding to antigen is activation of complement. So activation of complement, every antibody has complement binding sites on the constant region of the heavy chains. So this is the classical pathway of complement activation. When you have antibody bound to the microbe, that activates complement, and complement activates in a cascade manner. So once you activate one complement protein, you will have many proteins activated. And that will lead to opsonization, triggering of inflammation, and direct lysis of the microbe. Third benefit of antibody binding to antigen is opsonization. Basic definition of opsonization is when a microbe is coated with an antibody. In this case, when a microbe is coated with a protein, in this case, the protein is the antibody. When you have opsonization, it makes it much easier for the phagocytic cell to bind to the microbe and phagocytose it. Fourth benefit of antibody binding to antigen is neutralization. Neutralization means that when the antibody binds all over the virus, it prevents the virus from binding to cells, so that neutralizes the virus. It will not be able to infect the cells. If antibody binds all over a bacterium, it prevents it from adhering to the tissues of the human, and then that microbe can be flushed away or washed away. For exotoxins, if antibody is bound all over the exotoxin, then the exotoxin cannot affect the target cell. The last benefit of antibody binding to antigen is antibody-dependent cell-mediated cytotoxicity. And this is where the antibody binds to a eukaryotic microbe, and that signals to other white blood cells that they need to destroy this organism. So the antibody-dependent cell-mediated cytotoxicity, the antibody is basically tagging different organisms so that the other white blood cells know they need to eliminate it. The rest of the lecture is going to be over B cell development. And B cells are important because they are the white blood cells that actually produce the antibodies. So I'm going to talk about where B cells come from, how they mature, and how they become activated to actually start producing antibodies. B cells, like all leukocytes, they are derived from stem cells that are found in the red bone marrow. So that is where a B cell begins its life. It is derived from the stem cells and it is found in the red bone marrow. After the B cell is formed, the B cell has to go through maturation. And B cells mature also in the red bone marrow. And that is basically why B cells are named B cells, because it tells you where they are maturing. They are maturing in the red bone marrow. During maturation, there is a really important event that occurs, and that is called genetic shuffling. Genetic shuffling is an important event that occurs during the maturation of B cells. And the genes that are actually being shuffled and randomly selected are the genes that encode the variable region for the heavy chain and the variable region for the light chain. So genetic shuffling is going to determine the shape of the antigen binding site for the antibodies produced by a particular B cell. And the shape of the antigen binding site is going to determine what epitope is recognized. This image will help you understand genetic shuffling better. At the bottom is an image of an antibody. So here are the two identical heavy chains, here are the two identical light chains, and these colored regions represent the variable region of the heavy chain and light chain. So going through first the heavy chain, this is the variable region, and for the variable region there are actually three subregions. So a V region, which is represented by the red square, D region, which is by the blue hexagon, and a J region, the green circle. Every heavy chain variable region has one V region, one D region, and one J region. But for the V region, there are over 79 options for the structure of the V region. So there are 79 possible genes that you can select from to determine the shape of the V region. For the D region, there are 12 different genes, and you would select one. For the J region, there are 4 options for the genes for the structure of the J region. So during genetic shuffling, it is randomly determined which of these genes will actually encode for those regions. So for instance, for one particular B cell, maybe it is randomly determined that V4 will be selected. So V4 is selected to encode the structure for the V region, and maybe D2 is randomly selected to encode for the D region, and maybe J1 is randomly selected. So those are the 3 genes that are randomly selected to form the structure of the variable region. And once they are selected, they never change. Same thing happens for the light chain, except for the light chain there are only 2 regions. There is a V region with 100 options, and there is a J region with 5 options. So in this case, maybe V98 is selected, and maybe J5 is selected. So again, this is a random process. That's why it's called genetic shuffling. The shuffling implies that it is random. But once it is selected, then all the antibodies produced by that particular B cell will have exactly the same structure. Their antigen binding site will be exactly the same for both antigen binding sites on one antibody, and all the antibodies will recognize exactly the same epitope of an antigen. So the B cell arose from the stem cells in the bone marrow. It remained in the bone marrow to mature. During maturation, it went through genetic shuffling, which will determine which epitope the antibodies of this particular B cell will recognize. Once the B cell has finished maturing, then it is going to migrate to the lymph nodes and also the spleen. So all the B cells, once they have matured, they migrate out of the bone marrow and go to one of the lymph nodes or the spleen. And there they wait to be activated. So as I mentioned, once a B cell matures, it goes to the lymph node and it will wait to be activated. And on average, B cells will wait for about 3 months in the lymph nodes until they are activated. If they are not activated, then they will die off. But if they are activated, then they will go through the following process. So B cells, again, start from the stem cells in the bone marrow. They go through maturation. During maturation, they go through genetic shuffling, where it is determined what antigen their antibodies recognize. Once they have matured, then those B cells go to a lymph node. So here is an example of 4 different B cells that are waiting in the lymph node. And every B cell has a different antigen binding site on the antibodies. So remember, these antibodies, if they're in the cell membrane, they are IgD. So this B cell number 1, recognizes a round antigen. B cell number 2 recognizes a square antigen. B cell number 3 recognizes a triangular antigen, and B cell number 4 recognizes an inverted antigen, for instance. So these 4 B cells, along with actually thousands of other B cells, are waiting in the lymph nodes for an antigen to filter through, an antigen that entered the tissues and was flushed out of the tissues into the lymphatic vessels and is now being transported to the lymph node. So those B cells are waiting for an antigen that they can respond to. And B cell number 3 apparently has an IgD molecule that can recognize this particular antigen. Once a B cell comes in contact with the antigen it is specific for, then that B cell will be activated. And when a B cell is activated, it's going to go through a process called clonal expansion. So it is going to divide by mitosis to form daughter cells, genetically identical daughter cells. So that means all of the daughter cells are going to have exactly the same antigen binding sites as the original activated B cell. The first type of daughter cells that the activated B cell are going to form are memory cells. Memory cells are cells that are going to wait in the lymph node and will activate the second time the body comes in contact with this particular antigen. And these memory cells are going to wait for 10 years. If they have not been activated in 10 years, then they will die. So an activated B cell is going to go through clonal expansion, dividing by mitosis to form memory cells, which will wait for 10 years, and the other type of daughter cells will be plasma cells. And the vast majority of the daughter cells will be plasma cells. And the plasma cells are the cells that actually produce the antibody. So they're called plasma cells because they will be secreting the antibody into the plasma. And here the plasma cells are secreting antibody into the plasma, into the blood. And as you remember, the first class of antibody that they will be secreting is IgM. And after a few days, they're going to switch to IgG. So in this diagram, you can see that one particular B cell is able to make 3 different classes of antibody. So all the B cells start with IgD. And if they are activated, they can secrete IgM and then switch over to IgG. Related to your third line of defense is the idea of primary and secondary responses. A primary response is the first time you come in contact with a particular antigen, and the secondary response is the second time you come in contact with a particular antigen. Primary response. In the primary response, in terms of B cells, it is a naive B cell that is being activated. A naive B cell has never been activated before. That's what naive means, no experience. So in a primary response, it involves a naive B cell being activated. So with that naive B cell, the first class of antibody its plasma cells are going to produce is IgM. And later it will switch over to IgG a few days later. Also, this process takes a relatively long time. It takes about 10 days. And that's because it takes time for the antigen to enter into your tissues, to get flushed from the tissues, to go through the lymphatic vessel, to get to the lymph node, to get to a B cell that responds to it, and then those B cells have to go through mitosis. And for eukaryotic cells, mitosis can take between 18 and 20 hours. And once that happens, then the plasma cells have to start secreting the antibody, first the IgM, then the IgG. All of this takes time. So a primary response takes a long time. And relatively speaking, a primary response is fairly weak. But the important thing about the primary response is that at the end of this, you're going to generate memory B cells. So that's the important end product of the memory B cell, are the memory B cells, and they will wait around for 10 years in your lymph nodes. Secondary response. A secondary response is the second time you come in contact with a particular antigen. And if it is within those 10 years, you are now activating memory B cells. And most of the time it's more than one cell, because for every naive B cells, you generate about 50 memory B cells. And memory B cells are experienced. They've been activated before, so a memory B cell will right away start producing IgG and will still produce the IgM. So right away you have IgG being produced. And it takes a relatively short time, only about 2 to 3 days, to get significant levels of antibody being produced. And this is a very strong reaction because you have more than one B cell being activated, and you are producing IgG right away and high levels of IgG. Usually, a secondary response is so fast and so strong, and because you get IgG being produced, the IgG can go straight into the tissues to the site of infection right away. So because of all these things, usually secondary responses are able to clear the infection before you get signs and symptoms. So usually a secondary response is able to clear the infection before you get the disease. And when this happens, we say the person is immune. So if somebody is immune to a particular disease, that means that they can still be infected, but they'll have a secondary response which will be so fast and so strong it clears that infection before they get the disease. This is a diagram that shows you what is happening during a primary and secondary response. The x-axis is time in days, and the y-axis is the concentration, the level of antibodies in the serum. So at time 0, this is when the person is first exposed to the antigen, and there isn't any significant antibody production until about 10 days. And first to peak is the IgM, and then it takes a little bit longer till you get significant numbers of IgG. But at the end of this, you generate memory. Second exposure to the antigen. So this is the beginning of a secondary response. So sometime later, there's a second exposure to the antigen, and now you're going to have a secondary response, which is going to happen much more quickly. And first, IgG, you're going to get extremely high concentrations of IgG, and you still get the IgM being produced. But the high concentrations of IgG are going to clear the infection before you actually get disease. Adaptive immunity. Adaptive immunity is where a person develops the ability to be immune to a particular disease. And there are 4 types of adaptive immunity. Understanding adaptive immunity, you have to understand the 4 different terms that are involved in the definition. And the first 2 sets of terms are naturally acquired adaptive immunity and artificially acquired adaptive immunity. So natural and artificial. Natural means that this immunity is acquired in a natural way, part of the normal experience. Artificial means that there is medical intervention, which usually means there's some sort of injection. So those are the first 2 terms you need to know: naturally acquired immunity and artificially acquired immunity. The second 2 terms, the next 2 terms are active and passive. Active immunity means that the white blood cells are exposed to an antigen. They go through the process of activation, they go through clonal expansion, and they generate memory. Passive immunity involves just the transfer of antibodies. Naturally acquired active immunity is basically being naturally exposed to antigens, like being naturally infected, inhaling a respiratory microbe, ingesting a microbe, being bitten by a mosquito, having the microbe injected into your body. So in a natural way, your body is exposed to an antigen, and then it goes through a normal response. Passive immunity is a natural way where antibodies are transferred from one person to another. And the only natural process for that is from a mother to a fetus or a baby. So a mother can transfer antibodies to her baby in two different ways. IgG can cross the placenta and go into the fetus's circulation, and IgA can be transferred to the baby through breast milk. So this is naturally acquired active immunity, naturally acquired passive immunity. For artificially acquired immunity can be active or passive. Artificially acquired active immunity means that we are injecting antigens into the person's body to activate that person's own lymphocytes and generate memory. And an example of this is vaccination. So that's exactly what is happening during vaccination. Medical personnel are injecting antigens into the individual's body to generate memory. And then, of course, the idea with vaccination is that if the person is actually exposed to the pathogen, they will have a secondary response right away so that they can clear the microbe before they get the disease. Passive, artificially acquired passive immunity, this is where you are infusing antibodies into an individual. And usually this is in immunocompromised patients, people getting chemotherapy or have other immunocompromising conditions. And we take antibodies from other people and inject them into the person. The most common form of this is gamma globulin. Gamma globulin is IgG that has been acquired from other people. All right, so adaptive immunity has two types, naturally acquired immunity and artificially acquired immunity. And both of these have an active form and a passive form. The important thing about the active forms of acquired immunity is it's going to generate memory. Passive immunity, there is no memory because you've just transferred antibodies. And the immunity only lasts as long as the antibodies last. And IgG will last about 3 months. IgA lasts for about a month.